This method keeps biological samples in a native environment and reduces electron beam-induced artifacts. The main advantage is that this technique is applicable to whole cells, so cells don't need to be dehydrated, stained or sectioned before electron microscopy. The ability to image receptors on cancer cells in the context of the whole intact plasma membrane is a major aspect that can lead to new therapy approaches and the development of new anti-cancer drugs. I will demonstrate the work with cells, graphene coating and microscopy, and Sercan Keskin will show how to prepare the graphene.
To begin, add 100 microliters of cell suspension to 2 wells of a 96-well plate containing PLL and FLP-coated microchips, with the silicon nitride membrane facing up, and 50 microliters of serum-free medium. Incubate the plate at 37 degrees Celsius and 5% carbon dioxide for 5 minutes to allow cells to attach to the microchip. Check the density of the cells on the microchip with an inverted microscope, and make sure that the cells cover the window with sufficient space to flatten out and adhere. To label HER2 in the cells, place the microchips into the wells of the first row of the labeling plate. Then wash the microchips with PBS-BSA. To prevent unspecific binding of the antibody mimetic, incubate the microchips with PBS-BSA-GS for 5 minutes. Then with 200 nanomolar antibody mimetic for 10 minutes at 37 degrees Celsius and 5% carbon dioxide.